Welcome back. Next, we are going to hear from Dr. Amit Sachdev. Sorry, he is an Associate Chief Medical Officer at Michigan State University Healthcare. In addition to administrative duties, Dr. Sachdev is a neuromuscular medicine neurologist, MDA Care Center Director, and the physician to over 400 patients with MDA eligible diagnosis. Dr. Sachdev's twin passions are community building and new therapies. He is also the principal investigator on 6 clinical trials. So, Dr. Sachdev, I'll let you present and go ahead. You're quite busy. That's a lot of patients.
Thank you. Thank you so much, Nicole. Let me go ahead and share the presentation. Here we go. And Nicole, can you give me a thumbs up if everyone can see that?
It's perfect. Thank you.
Wonderful. Okay. So I must say it is an honor and a pleasure to join you today, and such an honor to join our very esteemed lecturers, both earlier today and coming up later, Dr. Wolf, Dr. Amato, Dr. Jelinas, and the many other folks who have come to talk today. I'm talking to you today about Myasthenia Gravis clinical trial update. And I understand that Dr. Wolf's lecture coming up next also speaks to some of the experimental drugs that are being used for myasthenia. So I thought that the best way to approach today's topic was to lay some groundwork into understanding where new treatment modalities sort of fit in the biology of the disease. And so we're going to try to paint the picture as to how these drugs work, and then the audience, the patient, you can then have some groundwork to figure out how this might fit into your particular scenario. So again, I'm the Associate Chief Medical Officer over at MSU Healthcare, and if you happen to be in Michigan, our clinic phone number is there on that slide. I do have some disclosures, and you can see that I do have clinical trial relationships with a number of partners, and I think this is a part of the reason why I was asked to give this talk. The relevant partners are Alexion, Catalyst, Argenx, Takeda, Momenta, and UCB. Those trials are all in the myasthenia space. There are also some relevant advisory boards and speakers bureaus, Alexion, Argenx, UCB. I'd like to include here that one of my disclosures is that I owe the U.S. federal government a ton of money because they hold all of my student loans. And that certainly does influence what I say about them. Additional disclosures, this was me when I joined MSU. I have less hair now, and I cannot function without my team. And so this is the team of physicians, nurse practitioners, nurses, physical therapists, ATPs for wheelchair support. These are the people that make care happen at Michigan State University. It's really a robust team and really some phenomenal providers there. The next thing I need to tell you about is that we are going to be talking about off-label and experimental use for medication, with the exception of eculizumab. Every medication that we discuss in this talk is either experimental or off-label, and that's particularly important to understand that none of this is FDA approved. And so when we talk about medicines that are not FDA approved, it's not that they can't be used. They can be used for the judgment of a physician, but it does mean that there is some opinion and art involved. So a quick roadmap of where we're going to go, and we should leave some generous time for questions here at the end. We're going to introduce myasthenia pathology. That's going to be just really quick about how myasthenia works. And the reason I'm going to do that is so that I can go into ways that we interfere with myasthenia. We're going to talk about T-cell therapies, B-cell therapies. We're going to talk about how do you interfere with antibodies getting to muscle, and then how do you interfere with the effects of antibodies when they're in the vicinity of muscle. And this is the way that our current medications work, but we're also going to introduce some up-and-coming concepts. Okay, so myasthenia gravis basics. Like I said, I'm not going to spend a lot of time here, just enough time to make sure that we're all on the same page. So this right here is a muscle, and on the outside of the muscle, you have a receptor. That receptor is the acetylcholine receptor, and the receptor is stabilized here in the wall of the muscle by some stabilizing proteins. And so for many patients with inflammation or autoimmune myasthenia, you have inflammation targeted to this apparatus, either directly to the receptor, that's 85% or so of our myasthenic, or to other areas around it, our striated antibodies here on the inside, our MuSK or agarine antibodies on the side, or on the nerve side of things, you have your Lambert-Eaton myasthenic syndrome, trouble releasing acetylcholine. And so this is a nerve, and nerves release acetylcholine so a signal will come down from the nerve will eject acetylcholine into this region between the nerve and the muscle. The acetylcholine will float on over and it will bind with the receptor and it will make the muscle twitch. And in myasthenia, there are antibodies. Those antibodies are produced by the body, they create inflammation, and they might block the receptor directly, they might target some of the stabilizing proteins, they might target the release of acetylcholine. But antibodies are a big part of how myasthenia happens. And so again, this is just an overview of the immunology of how myasthenia happens. We're not going to dwell further on this. We're going to talk next about how do you interfere with the process. Well, before we get into that, we're going to do a quick introduction on human clinical trials. So clinical trials are divided into 4 phases. And the reason I say human clinical trials is we certainly have other studies we do with drugs that form the trials of those medications. Those studies might involve petri dishes or animal models. But when we talk about human clinical trials, we talk about phase 1, often looking at safety, phase 2, often looking at what is the right dose, how is the best way to measure it, the effect of the drug. We think that these are often placebo controlled. That means that some patients get drug and some patients get a inert substance, a sugar pill, so to speak. They're often double blind. That means that both patient and doctor don't know what the patient is getting, but they often include an extension study. That means that after the double blind part is over, however many weeks that is, the patient is often given the opportunity to get the drug guaranteed for a period of time. Phase 3 is often larger. It's often a single dose. So in phase 2, we figured out what the right dose was. It's still placebo controlled. That means some people get drug and some people get an inert substance. It's still double blind and often includes an extension study as well. And then phase 4 is post-marketing. This is the drug is approved, but then there are some subgroups that might need additional study, perhaps studies in pregnancy, perhaps studies in more complicated cases, this sort of thing. So phase 4, the drug is available. Insurance companies are paying for it, but the company that has created the drug still needs to do additional work. So phase 3 is often the most exciting phase. At MSU, we have several phase 2 trials. We have some phase 3 trials. Phase 3 is often the pivotal phase. Okay, so thinking through therapies in myasthenia gravis. So we're going to start, and again, this is really geared towards the patient audience. So we're going to speak in some generalities. We're going to start with T cells. And the way this is going to work, we're going to put a slide up, and then we're going to put a figure up. And so the slide, we're going to give you some information, and we're going to paint the picture with the figure. So T cells come in a couple different varieties. You have T regulator cells, you have T killer cells, T helper cells. These often are thought of as cells that flip switches. that will form the barriers, for example, that allow you to attack yourself or no, or that will mediate how intense that attack is. And many of our classic drugs for myasthenia gravis, which are used off-label but are thought to work, including azathioprine, cyclosporin, mycophenolate, drugs that many of the patients on this call today might be taking, target T cells. So let's take a look at that. Let's go back to our diagram. So T cells are up here in this part of the diagram. And again, they come in the regulator variety or the helper variety or the killer variety. And so they influence the activity of inflammation that's happening down here at this junction. So we don't think that they quite are directly involved. They are more indirectly involved. And there are certain drugs that will influence their activity. This is azathioprine, cyclosporin, and mycophenolate. There is also a very early phase study on a new class of drugs that may change the activity of these regulatory cells. So the azathioprine, cyclosporin, and mycophenolate reduce the activity of this population of cells or damage the number of cells, of T cells that are available. So they generally reduce the number of or the intensity of activity of these T cells. The new class of drugs might, in fact, reinstate normal function of these cells. That new class of drugs affect tolerance, and that new class of drugs is some time away. This is very early animal study kind of data on drugs that affect tolerability. That is the ability of you to attack yourself. So traditionally, T cells has been the major focus of how we control myasthenia, essentially trying to reduce activity of the immune system by reducing the switches that turn it up or turn it on. And so trying to flip that switch back, new classes of drugs may attempt to hone the switch and turn it back more towards normal. Okay, so the next class of drugs is your plasma or your B cell oriented therapy. So what is a B cell or a plasma cell? These cells often produce antibodies. So the antibody is a protein that targets you or it targets an object that doesn't exist. So if you get the chicken pox, you never should get the chicken pox again. And that's not that you never get exposed to the chicken pox again. If you get the chicken pox while you're a child, you go on to have your own family, you have your own children get the chicken pox, you certainly care for that child, and you have several children, you get exposed again and again and again. And yet, you never develop the disease again. And the reason you don't develop the syndrome is because your body has created a defense. That defense comes in the form of antibodies. Antibodies are targeted, they're created by the immune system to target things that don't belong. In myasthenia, your body produces antibodies against either the nerve or the muscle, one of the two parts of the junction. And those antibodies are produced by plasma or B cells. And the antibodies have several different ways they interfere with the function of the junction. They can block the receptors from receiving their signal. They can cause the receptors to get taken in and destroyed, or they can cause inflammation at the site of where they bind. And there are drugs that target plasma or B cells. Rituxan is available off-label, also known as rituximab, and that targets the CD20 molecule on these groups of cells. And there is a new experimental drug that is currently enrolling in phase 2, that is the Takeda product, the Takeda 079 compound, which targets CD38 cells. Now, what you see here on the right is you see antibody levels. So this is how many antibodies you might have in your bloodstream. And as you can imagine, the drugs that target plasma cells or B cells are not very specific. So if you are used to living with a certain level of antibodies, and you go and you intentionally damage every plasma or B cell in the body, your antibody levels will drop. But that is all antibody levels, not necessarily just the ones that affect myasthenia. Now, there are some technical considerations here about certain subtypes of antibodies, the IgM versus IgG, the antibodies responsible for acute reactions versus chronic ones, the antibodies responsible for allergic reactions or parasite reactions. So those are some technical considerations. It's not necessarily that every single antibody type is depleted. But the bottom line is a dramatic reduction in antibodies occurs, and that reduction is not specific for just the antibodies that are causing myasthenia. And there is a slow recovery over time, but that recovery is measured in months. In fact, in many months, when you dose rituximab, you're measuring recovery at 6 months or more, some patients 12 months. And so this is a fairly blunt instrument, but it is one way, logically, of influencing myasthenia. And so let's look at our diagram. So what you see here is that these T cells do regulate the activity of the immune system, and they regulate these B cells over here, B cells and plasma cells. And these B cells or plasma cells produce antibodies, and that's what this little thing right here is. And these antibodies enter the bloodstream, that's what this is, and they arrive at the muscle and they attach to a target. Most of the time it's to the acetylcholine receptor. If you eliminate or damage this class of cell, you will drop your antibody levels by just producing less of them. But again, it will be in a very non-targeted way. And that's the same story over here with the T cells. The current medications we use, as thiopurine, cyclosporin, and Cellcept, decrease the function of T cells, but not in a very specific way. They reduce the function of T cells very broadly. And so there is some risk of getting infection. In both of these approaches that target the primary production of the immune response. So what if you could just prevent the antibodies from getting to muscle? If you could leave the infrastructure in place, but intervene before the antibodies get to the muscle, then you could leave some of your immune response, your ability to fight infection, while decreasing the intensity of the disease. And in fact, you can do that. So you can do that with plasma exchange, which many of our patients are on, intravenous immunoglobulin, or a new class of drugs, the FcRn Inhibitors. And so now I'm going to show you two graphs down here. Down here on your left, antibody levels over time, time measured in weeks, not in months. Plasma exchange draws those antibodies out of the bloodstream, but you're leaving the production of antibodies in place. So in weeks, the antibodies come back up. FcRn inhibitors, the new class of medications currently under investigation, do the same thing. Decrease the amount of circulating antibodies, but over the course of weeks, they start to trend back up. Now, with IVIG, it's a different story. With IVIG, intravenous immunoglobulin, and frankly, with subcutaneous immunoglobulin, it's the same story here, both of which, again, are used off-label for myasthenia. You have this sudden surge of antibodies in the bloodstream. This surge is because we are giving you other people's antibodies. But you can't hold on to all of those antibodies, and so you have this rapid destruction of the extra antibodies, and you go back to your level of normal antibody carriage. Whoops. So what is this in the red line? This is the number of antibodies you made on your own. So if you didn't do anything, the red line would just continue straight across. But you went and you flooded a person with antibodies, and then they destroyed all or a good chunk of the extra ones you gave them. And so the amount of antibodies that they produced on their own were destroyed in the process. A good chunk of the antibodies they made on their own were destroyed, but you left the infrastructure in place to produce antibodies still. And so slowly over time, the antibodies that the patient produces goes back closer to normal. So here the idea is, again, to sort of reiterate it, let's say you carry one antibody in your body and you only have the ability to carry one antibody. If you get 10, you now have 11. You cannot carry 11. You could only carry one. So very quickly, you degrade back to 1 because you can't carry 11. You can only carry 1. So the chances that you will keep the one you had is very small because now you have 11 for a short period of time. You can only carry 1. There's a greater chance that you're going to destroy what you had than keep it. And so this is playing the odds, flooding you with antibodies, and then a certain percentage of what you had before stays, but the vast majority of it goes. So again, depleting antibodies, that's plasma exchange and FcRn inhibitors. And so you have this sudden decline and then this gradual increase, and this is measured in weeks. And let's see how that looks. So again, T cells influence the activity of B cells. B cells produce antibodies. And in this situation, we're introducing plasma exchange. So what does that mean? We're introducing this needle. We're taking the blood out. We are putting a filter, and we're putting the blood back in. And so the antibodies are being captured in the plasma exchange machine. They're not being put back into the bloodstream. Here with FcRn inhibitors, what I've done is I've drawn the wall of the blood vessel. Now, the blood vessel is not just a pipe. The blood vessel is a living organ. Actually, I think, there we go. The blood vessel is a living organ. And because the blood vessel is a living organ, it is a part of the cleaning and metabolism of the blood it carries. And so a natural part of that process is that the blood vessel lining will take in parts of the blood constantly. It will destroy things that don't necessarily belong there, and it will spit back out things that do. So as a part of this process of taking part of the blood into the lining of the vessel, the antibodies will be taken in. The antibodies should have a path to then get spit back out, but they don't when you have this new class of medicines, the FcRn inhibitors. So instead, they get degraded. And the net result is that you don't carry any of your antibodies, or as many of your antibodies as you used to, and the antibodies don't get down here. Okay, so FcRn inhibitors is a very busy area of development. There are 6 companies actually involved, but 4 companies that are really at the phase 2 or phase 3 of testing. Those 4 companies are Argenx, UCB, Momenta, and Immunovant. And here are listed the names of the agents involved. My understanding of where these companies are with their phases of trials is listed in order here. So I believe Argenx has completed their myasthenia phase 3 and are now in FDA application, whereas UCB is still enrolling. Momenta has concluded, I think, phase 2 enrollment, and Immunovant is a little bit behind, I believe. Okay, so replacing antibodies, as we said, IVIG, we give patients a sudden surge of antibodies. They can't hold on to that. They degrade very quickly back to what they can hold. That degradation process results in a loss of antibodies that they had been producing on their own, but those antibodies go back to normal as they produce more over time. So how does that look? So again, this is this FcRn process. Here in black are the antibodies the patient produces on their own. Here in green are the antibodies that we flooded them with. The blood vessel lining takes in antibodies, but only has a certain capacity to spit them back out. And because you have so many more antibodies that you gave somebody versus antibodies they produced on their own, preferentially what gets spit back out is antibodies we gave them. Now, theoretically, we didn't give them any myasthenia antibodies because myasthenia is rare. And so the patient is degrading the antibodies they produced on their own, including the ones that drive myasthenia. Okay. Another common question, does IVIG manufacturer matter? There are many manufacturers. None of them are FDA approved for myasthenia syndrome. The major differences come in the form of concentration of the drug and the liquid that the drug is mixed in. So the liquid that the drug is mixed in is called a solvent, and some solvents are not logical in certain situations. For example, sugar water, dextrose, is not a logical solvent if you're diabetic. But for most patients, this is very personal. If you get a side effect burden with a particular variety of IV immunoglobulin, it makes a lot of sense to first try to slow the infusion down, but then to talk to your doctor about maybe switching the product altogether. Okay, we are almost done here. The next class of drugs is the complement inhibitors. So we've talked about how you can regulate the function of the immune system by addressing its master switches. Those are the T cells. Or you can regulate the function of the immune system by going after the factories that produce the antibodies. Those are the plasma cells or the B cells. Or you can allow the antibodies to be produced and then try to interfere with their ability to get to the muscle. That is plasma exchange, where you take the blood out, filter it, and put it back in. That is IVIG, where you flood the blood with antibodies, and then the body degrades some of them. Or that's FcRn inhibitors, where the body processes blood because the walls of the blood vessels are living tissues too. And that processing involves taking antibodies in along with a bunch of other stuff, getting rid of the bad stuff, and spitting the good stuff back into the blood flow. When you interfere with spitting antibodies back into the blood flow, you have a substantial reduction in antibody load. Complement inhibitors allow the antibody to make it all the way to the muscle. But one of the major focuses of what the antibody does when it's at the muscle is it creates inflammation and it attacks the muscle wall. it uses this chemical cascade called the complement cascade to create inflammation and attack the muscle wall. And a complement inhibitor prevents the inflammation from arising and prevents the attack, the insertion into the muscle wall of the terminal membrane attack complex, or the complement complex. So this is a lot like allowing a viral infection to sit in your nose, but taking a decongestant to avoid having the immune response to that viral infection affect your day-to-day life. So you allow the virus to be there, but you do not get the runny nose because you've taken a medicine to avoid that particular effect. With complement inhibitors, you allow the antibody to bind. We know there are several ways that the antibody might create damage. One of those ways is inflammation, and one of those ways is the membrane attack complex. The complement inhibitors inhibit both of those functions. So again, here's our diagram. We have the T cells, we have the B cells, we allow the B cells to create antibodies. Those antibodies make their way all the way over. They even attach to the receptors. Because they attach to the receptors, they create inflammation, and the complement inhibitor blocks the inflammation. So you avoid some of the damage to the muscle membrane because of the complement inhibitor. Okay, so what are the complement inhibitors on market today or in study? Alexion has two. The FDA-approved on market is eculizumab. This is the every 2-week infusion, and the trade name here is Solaris. Some of the patients in the audience may be on that drug. Then the other drug that they have currently in trial is ravulizumab, which is an every 8-week infusion. The Ra Pharma UCB Alliance has zilucoplan, which is a daily subcutaneous self-injection. This is a drug currently in phase 3. And so the complement inhibitors do have a member of this drug class, which is currently FDA approved, that is eculizumab. That is an important distinction between the complement inhibitors and the other types of medications that we've talked about, FcRn inhibitors, the drugs that focus on B cells or even T cells. None of those groups today have FDA-approved intervention. So to summarize, we have T cells, which come in regulatory, helper, and killer varieties. They influence the function of the immune system as it's attacking the nerve-muscle junction. You have drugs that are commonly used today, which restrict T cell populations. Because that's a very generic way to treat the immune system, you do open the window to certain complications. The next exciting class of agents may help return these T cells to more normal function, but that class of agents is very, very preliminary. And here is listed in one example. You have B cells. B cells produce antibodies. You can restrict the activity or damage the B cells, but again, that's another very blunt instrument. Those instruments last months in terms of their effect because you've gone and physically damaged the factories that produce antibodies. You generally should have a reasonable effect because antibodies drive this whole process, but it's not necessarily reversible. And there is one clinical trial agent in this group right here, that's the TACO-79. Because it is not as reversible as you would like, you have to wait for time for it to heal. You can allow the B cells to produce antibodies, but this is plasma exchange here where you take them out, filter them, and put the blood back in. You can prompt the blood vessel wall to degrade the antibodies by eliminating the ability to spit them back out. You can interfere with the ability of the antibody to produce inflammation, that is complement inhibitors. And so we're a little bit low on time here, but what we went over was clinical trial basics, an introduction to myasthenia pathology, T cells, B cells, and ways to interfere with the way antibodies get to muscle, and ways to interfere with antibody effects when they're in the vicinity of muscle. A little plug here, the Myasthenia Gravis Foundation of Michigan, where I am a board member, is going to be having its 44th annual meeting and conference. And here's some information on how you might be able to get more information about that. And with that, I believe that I have 6 minutes left for Q&A, which is what I was asked to do. Thank you so much.
That was right on the dot. Thank you. We have a person asking, where does Mestinon fall in these 3 types of drugs for myasthenia?
Sure. So let me go ahead and share the screen one more time. Actually, can you still see the screen?
No.
No. Okay.
You have to reshare.
Yeah. Let me reshare, and I will show you.
Okay.
What happened to maybe?
Did your
You know what?
icons go away?
Yeah, they did. Okay, so
hover, if you hover your mouse over, they might pop back up.
No, that's okay. So Mestinon does not treat to the underlying inflammation. So nerves release acetylcholine. Acetylcholine is supposed to float over, and it's supposed to bind to the muscle. And it binds at that receptor. Now, if you have a disease that damages the receptors, then you have less receptors available. And so how is it that you might get the receptors to still give you the sort of switch that you want it to? You might allow the acetylcholine to sit around longer and have an opportunity to bind more frequently. And that's what Mestinon does. So Mestinon allows the acetylcholine to sit in that area longer. And so if you don't have as many receptors as you should, Mestinon allows that acetylcholine to bind or bind several times, for example, to induce that twitch. Unfortunately, Mestinon does nothing to reduce the buildup of inflammation. And so you need often some other way to prevent the disease from getting worse. And so that's why oftentimes people are on prednisone or one of these other medications.
Mm-hmm. Can you explain why drugs that target T cells, like, I'm probably going to mess this up, azithromycin and is it mycopilate, takes several months to work in MG?
So it likely has something to do with the way that these drugs work.
Okay.
So we know that, for example, azathioprine prevents cells from dividing or interferes with cells making a copy of themselves. And so if you're slowing the process of making copies, you're not actually eliminating the copies that are currently present.
Okay. And
so that might be one reason why, is it just takes time for you to wait out the current copies diminishing in their numbers.
Okay.
But that is at best a guess. I would welcome Dr. Wolf's opinion as well, and his talk is coming up.
Yes, his is next. What factors would drive the type of complement inhibitor you would pick for your patient?
So today there is only one FDA-approved complement inhibitor, and so that drives the conversation completely today. One can envision in the future that there may be a reason to choose between multiple available options. Mm-hmm. The UCB raw pharmaceutical product, Zycloplan, is different from the Alexion product The Alexion products are antibodies. And so if you get an infusion of the Alexion product, it's supposed to sit around for a while. But if you then go and you use plasma exchange, or you use IVIG, or then you're interfering with the antibodies that are circulating in the patient's body, you're not capable of doing those two things at the same time.
Okay.
So if you had someone, for example, that was particularly brittle, you might choose dileucoplan, where that product is a different kind of protein and wouldn't get washed out if you gave somebody plasma exchange or if you gave them IVIG. And so that in the future might be a consideration, but today there's only one product. That product is eclusumab. And so today's consideration is eclusumab or a different type of therapy.
Okay. What is your view on removing the thymus?
I think that removing the thymus is very clearly supported by very strong data. And there are certain populations for which it is obviously the next rational step. And in particular, our younger patients. And it should be considered early in the disease course.
Okay.
It is my personal practice that I will want somebody well tuned up before I send them to the surgeon. And so I will take some time with a patient to make sure their disease is under good control so that they can tolerate the surgery better. But there are clearly patients who are very uncontrolled and go to thymectomy as a means to make their disease better. But thymus removal remains a major strategy for how we manage myasthenia gravis.
Okay, thank you. Someone's wondering, is the damage to the muscle receptors or muscle wall permanent in any situation?
So there are certainly patients who we think of as, quote, burnt out, where they have loss of muscle bulk, in particular up here in the deltoid.
Okay.
So those folks, we have a very hard time recovering their strength. But theoretically, that's really the minority. The nerve muscle junction should recover if you can manage the disease well. The other population for which we have a really hard time is our ocular myasthenic. Sometimes when someone has double vision, it can be very, very hard to get it back to normal. But for most patients, the expectation should be that we try to get you back closer to normal.
Okay. Sorry, we have two questions pop up. I just wanted to check the time. In theory, would the complement inhibitor class drugs be less effective over time, such as in relation to a form of a complement recovery, or would the effectiveness be consistent?
So it's very hard to know. The complement inhibitor drugs have been around for 10 or 12 years, and for the diseases that they were originally approved for, which are diseases of the kidney and diseases where patients have breakdown of blood, for those patients, they appear to work well even 10 or 12 years later. We just don't know. It's a different scenario for myasthenia gravis, where there's inflammation being driven rather than a genetic disorder.
Mm-hmm.
So we just don't know over time. The investigators who have had patients on these drugs the longest are about 6 or 7 years, and those investigators have reported to me when I've had conversations that patients seem to be doing well at that distance, but we just don't know.
Okay. And I believe this is our last question. This is an 80-year-old female MG, first diagnosed in 2008. She's had a crisis since January of 2020, double vision, upper body collapse. My upper body is bending forward. She's had IVIGs. It has taken up to 60 milligrams of prednisone, which helped my double vision, but now only on 20 because of its effect on my muscles. Would one of these new treatments help her?
Quite possibly. This is really going to be a scenario where you are going to need to talk with your treating physician. If you feel that you have not had a good relationship with your treating physician, you may want a second opinion. The bottom line is, I wouldn't be waiting for new treatment. You're in a scenario where your body is not functioning as well as it should today. And we have the ability to intervene on the disease today, perhaps not as elegantly as we would have in a few years. But many of the treatments we've discussed today are still pending FDA approval. And while promising, it is really important to understand that, in fact, most drugs that go into clinical trial, the odds of it failing are higher than the odds that the drug will actually get approved. And so I would not hold out for experimental medicine. I would take your best shot with a medication that we have available today if myasthenia is influencing your quality of life today.
And our last question, we know you talked a lot about thymectomies. Would you recommend a thymectomy if you have an MG antibody other than AChR, such as agrin?
So it is hard to say. And we did a poster on a patient who did well with a thymectomy and had an LRP4 antibody, but I have a very hard time saying that that would mean that everybody with other antibodies in myasthenia should have thymectomies. Again, Dr. Wolf might be able to offer a more concrete opinion here. If I recall correctly, he was intimately involved in the thymectomy trials. But I would really have to take that on a case-by-case basis. And that's what we did with our patient with LRP4.
Okay. All right. Well, thank you very much. I appreciate your time on a Saturday.
Of course. Thank you so much for having me.
All right. Thank you.